Convergent evolution, the process by which unrelated organisms independently evolve similar traits as a result of adapting to similar environments or selective pressures. Network motif, as defined by Uri Elon, a small set of recurring regulatory patterns in a network. Oscillations, repetitive variations of variables over time with a stable frequency or period. E-boxes, also known as enhancer boxes. These are short DNA regulatory elements in some eukaryotic promoters that act as binding sites for transcription factors and have thus been found to regulate gene expression. For clock-BMAL1, the consensus sequence for this cis-regulatory element is CACGTG. REV-DR2 and retinoic acid-related orphan receptor ROR binding elements ROREs. The cis-regulatory elements for the nuclear receptors REV-ERV and ROR. The consensus sequence of REV-DR2 RORE motifs involve RGGTCA half sites preceded by an AT-rich region. D-boxes, short cis-regulatory elements for the PAR-ZIP transcription factors DBP, D-box binding protein, HLF, hepatic leukemia factor, and TEF, thyrotroph embryonic factor. The common sequence of a D-box is TTATG-COT-AA. Phases of expression, the time of the day when peak gene expression occurs. Period, the length of the circadian rhythm measured from specific phase points in each cycle. For example, the peak-to-peak interval. Phosphoswitch, in the case of the PER2 protein, a phosphoswitch model is proposed whereby two competing phosphorylation sites, one for the FASPD site and one for the beta TRCP binding site, determine whether PER2 has a fast or slow degradation rate. Cystrome, the in vivo genome-wide location of transcription factor binding sites. Kamikaze model of transcriptional activation describes the ubiquitin-independent proteolysis of transcriptional activators, which suggests a role for activator degradation in RNA polymerase II elongation and the requirement for reloading of newly synthesized activators. Circadian time, CT, a standard of time based on the free running period of a rhythm oscillation. By convention, CT0 is the beginning of the subjective day and CT12 is the beginning of the subjective night. Global Run-On Sequencing (GROSEQ), a nuclear run-on assay method that captures nascent transcripts from initiated RNA polymerase II, followed by next-generation sequencing. Zeitgeber time (ZT), a standard of time based on the period of an environmental synchronizer or Zeitgeber, such as the 24-hour diurnal cycle of light and darkness. Under standard light-dark cycles, the time of lights on usually defines Zeitgeber time 0, ZT0, for diurnal organisms, and the time of lights off defines ZT12 for nocturnal animals. Nascent seq, next-generation sequencing of nascent RNA transcripts based on chromatin-associated RNA transcripts, nuclear run-on global transcripts such as GRO-seq or PRO-seq, or RNA sequencing of transcripts immunoprecipitated with RNA polymerase II. Laplacian analysis, a framework used in many disciplines to quantify topologies in which autonomous entities reach a consensus without a central direction. In the 4D nucleome, it is used to describe the underlying topology of genome-wide chromosome conformation capture interactions in the genome. Lamina-associated domains, regions of condensed chromatin that are bound by the nuclear lamina and are enriched for repressive histone marks such as H3K9ME2. Transcriptional architecture of the mammalian circadian clock. Abstract. Circadian clocks are endogenous oscillators that control 24-hour physiological and behavioral processes in organisms. These cell-autonomous clocks are composed of a transcription-translation-based autoregulatory feedback loop. With the development of next-generation sequencing approaches, biochemical and genomic insights into circadian function have recently come into focus. Genome-wide analysis of the clock transcriptional feedback loop have revealed a global circadian regulation of processes such as transcription factor occupancy, RNA polymerase II recruitment and initiation, nascent transcription, and chromatin remodeling. The genomic targets of circadian clocks are pervasive and are intimately linked to the regulation of metabolism, cell growth, and physiology. Energetic cycles governed by the solar day have driven the evolution of life on Earth. To adapt and respond optimally to the daily environmental cycles, living systems utilize internal timing systems that resonate with the 24-hour day. Across life forms, the internal timing system can be seen at the transcriptional level, giving rise to divergent gene networks in different organisms that oscillate on a 24-hour basis. Circadian clocks, as these endogenous timers are known, seem to be the product of convergent evolution, as the design principles of circadian transcriptional circuits share a common network motif, that is, negative feedback with delay, although the basic components are divergent across the kingdoms of life. The link to the daily solar energetic cycle is most obvious in photosynthetic organisms, but is also a conserved motif in the nutrient cycles of higher organisms. Whether circadian clocks evolved to adapt to the energetic demands of the environment or to avoid the detrimental consequences of solar radiation remains a topic of debate. Irrespective of why circadian clocks evolved, it is evident today that cell-autonomous clocks are ubiquitous. In mammals, for example, the neurocentric view of a master clock located only in the brain is no longer viable. Instead, the discovery of clock genes in the 1990s showed that virtually all cells express their genes and have the capacity to generate circadian oscillations. The circadian clock in mammals is now conceptualized as a hierarchical system in which a clock located in the hypothalamic suprachiasmatic nucleus acts as a master pacemaker to synchronize or entrain peripheral clocks distributed throughout the body. This system-wide architecture raises the following questions. How is this hierarchy coupled and integrated? And what are the functions of clocks in tissue-specific and cell-specific contexts? The molecular mechanism of the circadian clock in mammals is generated by a cell-autonomous transcriptional autoregulatory feedback loop. The core clock genes include clock and BMAL1, which encode activators, and PER1, PER2, CRY1, and CRY2, which encode repressors. Current topics of active investigation include the components of this core circadian clock gene network, how environmental and systemic inputs impinge on the core clock mechanism, and how the core clock network regulates downstream output pathways. Recent work has provided insight into the genomic targets of the core clock pathway and has led to some surprises concerning the pervasiveness of circadian regulation in gene expression. A substantial fraction, about 5 to 20%, of genes expressed in any particular cell or tissue have been found to undergo circadian oscillations at the mRNA level. However, this level of regulation, that is, steady-state mRNA rhythms, is only one of a multitude of layers of circadian regulation in gene expression. Virtually every regulatory stage in gene expression that has been examined in detail, including transcription, splicing, termination, polyadenylation, nuclear export, microRNA regulation, translation, and RNA degradation, has revealed additional layers of circadian control. Thus, both transcriptional and post-transcriptional regulation of circadian gene expression are key nodal points of control. Our understanding of the mechanism of the circadian clock in mammals has been informed by progress on two fronts. First, by the biochemical definition of components of circadian activator and repressor complexes, and second, by the structural determination of key domains of clock proteins. This review focuses on the molecular, biochemical, and genomic advances in our understanding of circadian regulation in mammals. I highlight work on the genomic targets on the core circadian transcriptional pathway, as well as studies revealing global circadian oscillations in RNA polymerase II, RNAP2 mediated transcription and their relation to the dynamic circadian regulation of chromatin state and chromatin modifiers. For non-mammalian model systems and aspects of circadian biology focused on photic regulation, metabolism, and immunity. There are other excellent reviews. The core circadian clock gene network. Core components of the mammalian circadian clock. In mammals, the mechanism of the circadian clock consists of the activator's clock and its paralog NPAS2 and its heterodimeric partner BMAL1, also known as ARNTL, which are basic helix loop helix BHLH per ARNT SIM path transcription factors. The clock BMAL1 complex binds to regulatory elements containing E-boxes in a set of rhythmic genes that encode the repressor protein period, encoded by PER1, PER2, and PER3, and cryptochrome, encoded by CRY1 and CRY2. In mice, clock BMAL1 activation occurs in the daytime, leading to the transcription of the purr and cry genes in the afternoon and the accumulation of the purr and cry proteins in the late afternoon or evening. The purr and cry proteins interact with each other as well as with the serine threonine kinases, casein kinase 1 delta, CK1 delta, and CK1 epsilon, and translocate into the nucleus at night, where they interact with clock BMAL1 to repress their own transcription. As repression progresses, purr and cry transcription declines, and the purr and cry proteins levels decrease. The half-lives of purr and cry proteins are relatively short, and specific E3 ligase complexes target these proteins for ubiquitination and subsequent degradation by the proteasome. Once negative feedback depression is relieved by turnover of the repressor complex, transcription by clock BMAL1 can begin anew to start a new cycle of transcription the next morning. Importantly, core clock genes, such as those that encode purr2 and its regulatory kinase CK1 delta, have been found to underlie human sleep timing disorders, thus demonstrating a clear role for clock genes in humans. In addition to the purr and cry target genes, the clock BMAL1 complex also activates the nuclear receptors Rev-ERB alpha and Rev-ERB beta, encoded by NR1D1 and NR1D2 respectively, which compete at Rev-DR2 and retinoic acid-related orphan receptor ROR binding elements ROREs, with ROR alpha, ROR beta, and ROR gamma encoded by RORA, RORB, and RORC respectively. The rhythmic expression of Rev-ERB alpha and Rev-ER beta leads to the repression of BMAL1 and clock, which induces a rhythm in these genes that is in an antiphase with purr expression rhythms. These interlocked feedback loops of activators and repressors form a canonical positive feedback and negative feedback gene network motif. A third clock BMAL1 driven transcriptional loop involves the PAR-B-zip, proline and acidic amino acid rich basic leucine zipper factors DBP, D-box binding protein, TEF, thyrotroph embryonic factor, and HLF, hepatic leukemia factor. These proteins interact at sites containing D-boxes with the repressor NFIL3, nuclear factor interleukin-3 regulated, also known as E4BP4, which is driven by the Rev-ERB-ROR loop. Together, These three interlocking transcriptional feedback loops can generate cycles of transcription with various phases of expression depending on the combination of cis elements, that is, E boxes, ROREs, and D boxes, in the promoters and enhancers of specific target genes. Core clock protein interactions with chromatin and chromatin modifying complexes. At the beginning of the transcription cycle, the activators clock and BMAL1 interact with the histone acetyltransferases HATs P300 and CREB binding protein CBP respectively to acetylate histones and provide an accessible chromatin state for transcription. Clock has been reported to have intrinsic HAT activity and to acetylate histone H3 and lysine 9, H3K9, and lysine 14 residues H3K14. The NAD plus dependent histone deacetylase HDAC sirtuin 1 associates with clock BMAL1 and PER2, and a circadian rhythm in NAD plus levels driven by the expression of the clock BMAL1 target gene NAMPT in turn leads to a rhythm in SIRT1 activity that feeds back to inhibit the clock BMAL1 complex. Clock and BMAL1 have also been shown to be associated with the methyltransferase MLL1 mixed lineage leukemia 1. Interestingly, MLL1 interacts with the delta 19 activation domain of clock and is thought to be important for the recruitment of clock to DNA. Moreover, MLL1 is associated with rhythmic H3K4 trimethylation, H3K4ME3, a common histone marker associated with active gene promoters. In addition, MLL1 is acetylated and the metabolic regulator SIRT1 rhythmically deacetylates MLL1, thereby affecting its methyltransferase activity. The related methyltransferase MLL3 exhibits circadian occupancy and is associated with promoters bearing rhythmic H3K4ME3 marks. However, MLL3 recruitment to DNA does not depend on BMAL1 or clock. Clock and BMAL1 also interact with the lysine-specific histone demethylase JARID1A, JARID1A, and LSD1, which are both thought to promote the recruitment of clock BMAL1 to PER promoters to enhance transcription. BMAL1 interacts with TRAP150, thyroid hormone receptor associated protein 150, a co-activator that promotes clock BMAL1 binding to circadian target genes and links clock BMAL1 to the mediator complex subunit MED1 to recruit the general transcriptional machinery. RNA polymerase II is both recruited and initiated on a circadian basis, as discussed in more detail below. During the transition from activation to repression, the clock BMAL1 complex interacts with the per-cry repressor complex at the beginning of the night, as the levels of per and cry accumulate and translocate to the nucleus. The per complex is large and contains at least 25 proteins. Initially, clock and BMAL1 are associated with two components of the MI2 nucleosome remodeling and deacetylase NURD transcriptional co-repressor complex, HCD4 chromodomain helicase DNA binding protein 4, and MTA2 metastasis associated one family member 2. Interestingly, per complexes without clock BMAL1 lack HCD4 and MTA2 but contain other members of the NURD complex, such as MBD2, methyl CpG binding domain protein 2, GATAD2A, GATA zinc finger domain containing protein 2A, HDAC1 and RBAP48, retinoblastoma binding protein P48. This finding indicates that during this transition phase, there is a split NURD complex that then comes together to form a reconstituted NURD co-repressor complex to inhibit clock BMAL1 transcription. The clock BMAL1 complex also recruits polycomb repressive complex 2, PRC2, leading to the trimethylation of A3K27 at target genes and thereby inhibiting gene expression. In addition, clock BMAL1 recruits the DNA damage binding protein 1, DDB1, tulin 4, CUL4, ubiquitin ligase complex to circadian target genes to promote H2B monoubiquitination at EBOX sites to covalently mark genes for subsequent recruitment of the per complex. At the peak of repression during the middle of the night, the per complex is also associated with the transcriptional repressor complexes SIN3HDAC and HP1 gamma SUV39H histone methyltransferase, which contribute to clock BMAL1 repression through the deacetylation of histone A3K9 and H4K5, as well as demethylation and trimethylation of A3K9. The per repressor complex also includes NONO, a protein involved in RNA processing, and WDR5, a component of a histone methyltransferase complex. At later times during the repression phase, PER complexes include the RNA helicases DDX5 and DHX9, as well as the large subunit of RNA polymerase II and SETX, a helicase that promotes transcriptional termination. Thus, the recruitment of several chromatin-modifying complexes as co-repressors is necessary for PER-mediated and probably CLY-mediated repression of clock BMAL1. During the circadian cycle, transcriptional activation and repression are accompanied by dynamic epigenetic transitions in chromatin state that poise the genome for rhythmic gene expression. Regulation of circadian period length by repressor stability. The stability of the core repressor proteins is an important determining factor in the dynamics of the transcriptional feedback loop, which in turn regulates the period length of the oscillation. The stability of PER and CRY is regulated by their phosphorylation state and by their ubiquitination by E3 ligase complexes. For example, the tau mutation in CSNK1E, which encodes CK1Y, shortens the circadian period length in vivo. Moreover, this mutation causes the destabilization of PER proteins by phosphorylating a regulatory site that marks the PER proteins for ubiquitination by beta-TRCP, also known as FBOXWD, repeat containing protein 1A, and proteasomal degradation. By contrast, mutation in the gene encoding the FBOX protein, FBXL3, which is one of 4 subunits of the SCF, SKP1 Culin FBOX protein, E3 ligase complex, lengthen the circadian period and stabilize the CRY proteins by decreasing ubiquitination and subsequent proteasomal degradation. In addition, CRY ubiquitination by SCF, FBXL3, is triggered by 5' AMP activated protein kinase, AMPK-mediated phosphorylation, which provides a link to metabolism via nutrient-responsive AMPK activity. The stability and turnover of PER proteins is primarily regulated by several mechanisms, including by CK1 delta and CK1Y, by the balance between these two protein kinases and phosphatase 1, and by the stoichiometry of PER proteins and these kinases. Recent work has shown that a dual kinase, multisite phosphoswitch, regulates PER stability and its ubiquitination by the beta-TRCP E3 ubiquitin ligase complex. For cry proteins, their stability and turnover are regulated by the balance of the two paralogs FBXL3 and FBXL21. Contrary to expectation, FBXL21 antagonizes FBXL3 to reduce the E3 ligase activity of SCF FBXL3 on cry1 and cry2 to stabilize them. Although SCF FBXL21 does possess E3 ligase activity, it is less efficient than SCF FBXL3 in the ubiquitination of cry. However, FBXL21 interacts more strongly with cry than FBXL3, which protects cry from SCF FBXL3 activity. The subcellular localization of FBXL3 and FBXL21 also differ, with FBXL3 acting as the primary E3 ligase for cry in the nucleus, whereas FBXL21 acts as the primary E3 ligase for cry in the cytoplasm. Finally, in genetic experiments exploring the interaction of the CK1Y tau mutant with the FBXL3 AFH mutant, a broad range of periodicities were seen depending on the combination of alleles, indicating that the PER and cry pathways independently affect the ultimate period length in vivo. Genome-wide clock architecture. Although previous work has examined the transcriptional regulation of clock BMAL1 target genes such as DBP, PER1, and NR1D1 in detail, the identity of the circadian syndrome has only been recently defined. Using chromatin immunoprecipitation followed by sequencing ChIP-seq, several research groups have identified genome-wide DNA binding sites for BMAL1, clock, NPAS2, PER1, PER2, cry1, and cry2 in mouse liver during the circadian cycle. Genomic targets of the core circadian activators. Occupancy of clock BMAL1 at the DBP locus, which encodes a transcription factor that modulates the expression of clock output genes, is circadian, with peak binding occurring during the middle of the day. At the single cell level, clock and BMAL1 recruitment to E-boxes in DBP are interdependent and circadian. BMAL1 binding is dynamic and stochastic, and fluctuations in binding depend on the proteasome, which is consistent with a kamikaze model of transcriptional activation. On a genome-wide level, clock and BMAL1 occupancy also peak within a narrow window of time during the middle of the day, and the phasing of clock BMAL1 occupancy is not tightly phase-locked with the RNA expression of their target genes. This flexible phase relationship can be partially explained by the combinatorial action of other cis-acting elements, ROREs and D-boxes, within the tripartite clock gene network. For example, CRY1 expression, which peaks much later in phase the PER genes, is regulated by both E-boxes and ROREs. In an effort to view the dynamics of the occupancy of the core circadian activator complexes in a genome-wide context, one study assessed all core clock proteins in the mouse liver. At PER1, the activators BMAL1 and clock bind in a cyclic manner at the promoter between circadian time 0, CT0, and CT12, with maximal binding observed at CT8. In genome-wide analysis, clock and BMAL1 bind to more than 4,600 and 5,900 sites, respectively, corresponding to about 3,000 unique genes in the liver. At highly expressed target genes, clock BMAL1 DNA binding motifs are enriched for a tandem E-box motif. Native liver extracts showed that clock BMAL1 bind to tandem E-box sites cooperatively with higher affinity to more effectively compete with other E-box binding factors such as USF1, upstream stimulatory factor 1. The hepatic steroid receptor coactivator 2, SRC2, also known as NCoA2, is recruited to DNA in a diurnal cycle and overlaps extensively with clock BMAL1 sites. SRC2 acts as a potent transcriptional coactivator of clock BMAL1, and null SRC2 mutations in mice lead to an abnormally early phase of the activity rhythm, indicating that SRC2 has a role within the circadian clock system. In addition to the liver, the circadian syndrome has recently been described in mouse pancreatic islets. In a comparison of the occupancy of BMAL1 between the liver and the pancreas, about 40% of the target genes overlapped, with disparate binding sites clustering at distal regulatory regions of tissue-specific genes. Interestingly, even within the overlapping set of target genes, BMAL1 was found to bind to distinct enhancers in the liver and pancreas, consistent with the existence of tissue-specific circadian cis-regulatory modules. Genomic targets of the core circadian repressors. In contrast to the activators, the repressors PER1, PER2, and CRY2 bind genome-wide at night between CT12 and CT20, peaking between CT15 and CT17. CRY1 has a bimodal pattern, with a major peak at CT0. The repressors CRY1 and CRY2 bind to significantly more sites in the genome than PER1 and PER2, with many thousands of these sites being independent of clock BMAL1 and containing DNA binding motifs for nuclear receptors. Consistent with this finding, CRY1 interacts with several nuclear receptors and is a co-repressor for the glucocorticoid receptor. The nuclear receptors REV-ERB alpha and REV-ERB beta also occupy DNA rhythmically, with a peak in the daytime. REV-ERB alpha competes with ROR alpha for DNA binding at RORE and REV-DR2 sites to regulate circadian target genes such as BMAL1 and CRY1, where it recruits nuclear receptor co-repressor 1 and CoR and HDAC3 to repress gene expression. Interestingly, REV-ERB alpha also interacts at other genomic sites that are enriched for tissue-specific transcription factors such as hepatocyte nuclear factor 6, HNF6, in the liver. At these sites, the action of REV-ERB alpha is independent of its DNA binding domain and defines a different set of target genes that are directly involved in metabolism. Finally, SRC2 has been proposed to modulate an oscillation in chromatin accessibility by recruiting PBAF, polybromo associated BRG, Brahma associated factors, members of the SWI-SNF switch sucrose non-fermentable complex, to promote REV-ERB loading and enhance the amplitude of circadian gene expression. Thus, the circadian system is highly dynamic and involves the core circadian E-box transcriptional regulators and is interlinked via REV-ERB alpha or beta, as well as DBP and NFIL3 to secondary and tertiary gene targets with a wide range of phases of gene expression. Genome-wide circadian RNA expression. To examine the functional consequences of the circadian system, RNA sequencing, RNA-seq, has been used to profile cycling genes in the mouse liver. For example, using both the intron RNA signal as a proxy for pre-mRNA and the exon D RNA signal as a proxy for mRNA. Strand-specific whole transcriptome RNA-seq revealed a robust circadian oscillation at the DBP and PER2 genes. As was previously observed in microarray experiments, thousands of genes cycle at the steady-state mRNA level in the liver, as well as in many other tissues. As more regions in the mouse have been profiled, more than half of all genes have been found to cycle at the mRNA level in at least one tissue. Importantly, the identified cycling genes are enriched for drug targets. More than half of the top-selling 100 drugs on the market in the United States have a circadian target gene. This finding emphasizes the importance of optimizing the timing of drug administration for circadian drug targets, which can both enhance efficacy while at the same time reduce side effects. A wide range of cycling RNA species has been found using RNA-seq, in addition to cycling protein-coding transcripts, oscillating long intergenic non-coding RNA, microRNA, and antisense RNA transcripts have been found in the mouse liver. Using global run-on sequencing, GROW-seq, to identify enhancer RNA, eRNA transcripts. Thousands of oscillating enhancers have been found, and the phase of these cycling eRNAs corresponds with the phases of transcription of nearby genes. A motif analysis of cycling enhancers at different circadian phases revealed that there was phase-specific enrichment of transcription factor motifs, and that 4 sets of motifs were enriched at different times. E-box motifs were enriched between Zeitgeber times 6, ZT6, and ZT9, corresponding to the peak of clock BMAL 1 occupancy. D-box motifs were enriched between ZT9 and ZT15, corresponding to the expression of DBP and the low point of N-fil3 repression. RORE and REV-DR2 motifs were enriched between ZT18 and ZT24, corresponding with the low point of REV-ERB repression of the RORs. ETS E26 transformation specific binding motifs were enriched between ZT0 and ZT3, which may correspond to GM129, also known as circadian associated transcriptional repressor or chrono repression. Overall, the circadian systromic results agree well with their transcriptional readouts as assessed by eRNA and gene expression analysis. The tripartite core circadian transcriptional feedback loop, E-box, D-box, and RORE, drives the majority of circadian gene expression on a genome-wide level. Using whole transcriptome RNA-seq or nascent seq combined with mRNA-seq, two divergent sets of cycling genes have been found. Cycling nascent transcripts and cycling mRNA levels. Only about 20% of cycling mRNAs also had a corresponding cycling nascent transcript, indicating that other RNA processes are likely to be contributing to steady-state circadian mRNA expression. Several different classes of cycling transcripts have been observed, including the following. First, cycling transcription and cycling steady-state mRNA. Second, cycling transcription and no cycling mRNA. And third, no detectable cycling transcription but cycling mRNA levels. The first class is typical for the core circadian genes and their high amplitude direct targets. The second class is typically seen in highly expressed genes in the liver that have mRNAs with long half-lives, such as albumin. The third class represents transcripts that could become rhythmic via circadian variation in mRNA turnover, RNA splicing, polyadenylation, microRNA regulation, and other post-transcriptional steps. Whether rhythmic translation or protein levels ultimately occur has also been revealing. For example, rhythmic diurnal polyadenylation occurs in 2.3% of expressed genes in the liver, and 1/5 of these genes, third class, lead to rhythmic protein expression that correlate with poly-A tail length in the absence of rhythmic mRNA levels. Ribosomal profiling has also revealed substantial numbers of examples of circadian translation in U2OS cells and liver cells. Interestingly, there are at least 150 examples of translationally driven oscillations in the liver for which no mRNA rhythms are present. Thus, both transcriptional and post-transcriptional regulation are critical for circadian gene expression. Genome-wide circadian regulation of transcription, RNA polymerase II, and chromatin states. In the mouse liver, cycling pre-mRNA levels using the intron RNA signal were coordinately expressed in time, with a peak at CT15. Thus, there is a peak of globally coordinated circadian transcription that occurs at the beginning of the night during the active or awake portion of the mouse circadian rhythm, which is not seen at the mRNA level, which has a bimodal pattern. To further analyze the global rhythms in nascent transcription, RNA polymerase II occupancy has been measured during the circadian cycle. The large subunit of RNA polymerase II, POLR2A, contains a carboxy terminal domain, CTD, that is post-translationally modified at different stages of transcription. RNA polymerase II is recruited into the pre-initiation complex in a hypophosphorylated state, recognized by the 8WG16 antibody. Surprisingly, RNA polymerase II AWG16 occupancy is highly circadian in the mouse liver, with a peak that correlated with the intron RNA peak. The first step in the initiation of RNA polymerase II involves phosphorylation of SIR5P of the CTD of RNA polymerase II. RNA polymerase II SIR5P occupancy is also circadian, indicating that both recruitment and initiation of RNA polymerase II are under circadian control. An antibody against the RPB2 subunit of RNA polymerase II, POLR2B, revealed that RNA polymerase II occupancy for promoters and gene bodies is rhythmic and co-varies over time. Rhythmic loading of RNA polymerase II to promoters rather than a rhythmic transition from a paused state to productive elongation accounts for the majority of rhythmic transcription in the mouse liver. Given these circadian rhythms of RNA polymerase II, chromatin states associated with RNA polymerase II transcription have been examined during the circadian cycle. Figure 5 illustrates genome-level views of histone modifications that are characteristic of promoter, enhancer, and elongation states. Histone H3K4ME3, H3K9AC, and H3K27AC modifications are enriched at promoters and undergo circadian rhythms in histone modifications at per1. On a genome level, circadian rhythms in RNA polymerase II occupancy, as well as histone H3K4ME3, H3K9AC, and H3K27AC modifications are present in the majority of expressed genes irrespective of whether RNA cycling occurs. Both RNA polymerase II occupancy and A3K4ME3 marks are highly dynamic and show circadian oscillations that peak during the nearly night CT12 for all expressed genes cycling intron RNA genes and non-cycling genes. In summary, in the mouse liver, there is a highly stereotyped, time-dependent pattern of core transcription factor binding, RNA polymerase II occupancy, transcription and chromatin states. There are 6 distinctive phases of circadian cycle. 1. A poised state at the beginning of the cycle, CT0, during which clock and BMAL1 and CRY1 bind to E-box sites in a transcriptionally silent state. 2. A de-repression phase at CD4 as the levels of CRY1 decline. 3. A major activation phase at CT8 during which clock BMAL1 occupancy peaks along with P300 and CBP recruitment and are accompanied by histone H3K4ME1 and H3K9AC activation marks. 4. A temporally coordinated transcriptional activation phase at CT12 to CT15 during which RNA polymerase II recruitment and pre-mRNA transcript expression peak, followed by histone A3K4ME3 and A3K27AC marks. 5. A classic repression phase at CT15 to CT18 during which PER1, PER2 and CRY2 occupancy peaks and transcription declines. And 6. A transition between the end of repression and the beginning of the next cycle. Interestingly, the co-activator CBP interacts with the repressor PER2 at the end of the repression phase, and CBP occupancy has a second peak at this time, perhaps maintaining an open chromatin state during the transition from the repression to activation. CRY1 occupancy also bridges this transition period and interacts with clock BMAL1 to begin the next cycle. In contrast to embryonic stem cells, for which 30% of genes are paused and carry bivalent histone marks, genes that are not expressed in the mouse liver are not enriched for paused RNA polymerase II. Interestingly, the histone H3K4ME3 promoter mark lags RNA polymerase II occupancy by approximately 1 hour in cycling genes, and the H3K36ME3 elongation mark lags by approximately 3 hours, consistent with the idea that these marks arise as a consequence of email. consistent with the idea that these marks arise as a consequence of RNA polymerase II transcription. Thus, circadian transcriptional regulators are clearly involved in RNA polymerase II recruitment and initiation, as are the histone modifications associated with these events. However, additional steps such as promoter proximal pausing, pause release, and productive elongation probably have additional regulatory roles. At enhancer sites, ClockBMAL1 is a pioneer transcription factor and can bind nucleosomes to promote rhythmic chromatin remodeling and incorporation of the histone variant H2A.Z. Cycling enhancers also display circadian rhythms in eRNA expression in conjunction with tissue-specific transcription factors. In the case of cycling genes, RNA polymerase II recruitment, initiation, and productive elongation are all rhythmic and occur in phase, leading to associated rhythms in histone modifications. In the case of expressed but non-cycling genes, RNA polymerase II recruitment and initiation are also rhythmic, leading to rhythmic histone modifications at the promoter. However, subsequent steps in pausing, pause release, and productive elongation may no longer be under circadian regulation, because RNA polymerase II pausing at promoter proximal regions and pause release are major regulatory steps in transcription. It will be important in the future to determine whether pausing factors such as negative elongation factor (NELF) and DRB sensitivity inducing factor (DSIF), as well as the positive transcription elongation factor B (PTEFB) complex are under circadian control. Conclusions. The circadian modulation of transcription occurs on a genome-wide scale far greater than that seen previously by gene expression profiling. The circadian clock in the liver regulates RNA polymerase II occupancy on a genome-wide basis and drives the genome-wide circadian modulation of chromatin states. These circadian rhythms in the chromatin state poise the genome for transcription on a daily basis to coincide with the daily metabolic demands of the organism and to optimize the energy utilization of gene expression. In addition to the circadian modulation of chromatin states, the 3D architecture of the nucleus and the interaction of active and repressive chromosomal domains undergo circadian oscillations. Thus, the four-dimensional regulation of the genome and its nuclear architecture are ripe for study. In addition to the pervasive circadian regulation of metabolism, strong links between the circadian gene network and cell growth and cancer. Immune function and signaling are apparent, and components of this network will be important therapeutic targets in the future. Figure 1. Circadian rhythms are adaptations of organismal physiology to resonate with the daily solar energetic cycle on Earth. A. The Earth's 24-hour rotation leads to a diurnal cycle of light and darkness that drive an energy harvesting and energy storage daily rhythm. Solar irradiation also imposes a cycle of DNA damage and recovery. B. Animals have behavioral rhythms of sleep-wake, and feeding and fasting occur on a 24-hour basis in synchrony with the solar day. C. A conserved network motif of circadian clocks involves a transcription translation negative feedback loop with delay. D. In mammals, circadian clocks are cell autonomous and are found in all major organ systems and tissues of the body. A hierarchical organization exists in which the hypothalamic suprachiasmatic nucleus, SCN, acts as a master pacemaker to synchronize behavioral and physiological rhythms throughout the body. RE, regulatory element. Part A and Part B are reproduced from REF. Reference 11. Box 1. Structural biology of clock proteins. Structural biology will have an essential role in achieving a mechanistic understanding of clock function. The 3D structures of several circadian proteins and their complexes have been solved using X-ray crystallography. These structures include the basic helix-loop helix, BHLH, Pass A and Pass B domains of the heterodimeric transcriptional activator complex, clock BMAL1, protein data bank identifier 4F3L. The Pass A and Pass B domains of the mammalian period, per proteins. The photolyase homology region, PHR, of the mammalian cryptochrome 1, Cry1, the PHR of Cry2 in complex with the F-box protein, FBXL3, and the Per2CBD Cry binding domain in complex with Cry1 or Cry2. Protein data bank ID 4U8H. The structure of the clock BMAL1 complex is asymmetric, with clock wrapping around BMAL1, The path A and path B domains of the two subunits interact with their complementary domains, but with two different interfaces. The path A domains have symmetrical interactions involving the beta seed surfaces, whereas the path B domains have a head-to-tail interaction in which the beta sheet surface of BMAL1 interacts with the alpha helical surface of clock. The path A and path B domain configuration of BMAL1, but not clock, is identical to that seen in the path A and path B domains of the mammalian PER proteins, indicating that PER could interact with clock and replace BMAL1. Interestingly, a crystal structure of another member of the BHLH PAR ARNT SIM path heterodimeric protein family HIF2 alpha, also known as EPAS1 ARNT, reveals a completely different overall domain arrangement compared to clock BMAL1. However, specific domain interface interactions are preserved between the respective BHLH path A and path B domains of the two subunits. The recognition of the CACGTG E-box DNA binding motif of the clock BMAL1 BHLH region is similar to that seen for other BHLH proteins. The crystal structures of mammalian Cry2 have been determined in 5 functional states. 1. The PHR by itself, apple. 2. The FAD bound Cry2 PHR. 3. The Cry2 FBXL3 SKP1 complex. 4. The KL001 bound Cry2 PHR. And 5. Cry2 in complex with PER2 CBD. Both the Cry1 and Cry2 apple structures adopt the canonical photolyase fold and can be aligned most closely to the Drosophila photolylase. However, unlike 6,4 photolysases and cryptochromes from plants and Drosophila melanogaster, the mammalian Cry1 and Cry2 PHRs do not have the FAD cofactor bound. In Cry2, FAD can bind with low affinity, and the FAD binding pocket has a more open conformation, perhaps explaining the lower affinity for FAD. Cry2 in complex with FBXL3 reveals an extensive interface with the FBXL3 leucine-rich repeat LRR domain. Moreover, most interestingly, the carboxy terminal tail of FBXL3 inserts into the FAD binding pocket, revealing a novel substrate recognition motif by the SCF ubiquitin ligase. The small molecule stabilizer of Cry, KL001, binds to the FAD pocket, but also extends beyond the pocket where the FBXL3 C-terminal tail enters. This binding therefore blocks the FBXL3 Cry2 interaction to stabilize Cry. Finally, the known interaction between Cry and PER proteins, which mutually stabilize one another, has been illuminated by the recent crystal structures of the Cry1 PER2 CBD and Cry2 PER2 CBD complexes. The PER2 CBD forms an extended conformation on Cry2 that begins at a secondary pocket critical for clock BMAL1 binding and winds around the Cry2 C-terminal helix to sterically hinder the recognition of Cry2 by the SCF FBXL3 ubiquitin ligase. Thus, the overlapping interfaces on Cry2 for PER2 CBD and FBXL3 provide a structural basis for the competition of PER proteins and FBXL3 for controlling the stability of Cry. The existing crystal structures raise the question of how PER and Cry interacts with clock BMAL1 as a quaternary complex. In different contexts, Cry1 can interact directly with clock BMAL1 without PER in vitro, although in other experiments PER has been shown to be essential for interaction with clock BMAL1 in vivo. It seems probable that PER is involved in reducing the occupancy of clock BMAL1 on DNA, as seen in Drosophila melanogaster, whereas Cry1 may be able to repress clock BMAL1 on DNA, because the native clock BMAL1 PER Cry quaternary complexes are megadalton in size and involve other interacting proteins, and important domains of these proteins are intrinsically disordered. The solving of these complexes will probably require a combination of crystallography, NMR, and cryo-electron microscopy methods. Figure 2. The circadian gene network in mammals. At the core of the network, the basic helix loop helix BHLH per ARNT SIM path, transcription factors clock and BMAL1 activate the PER1, PER2, Cry1, and Cry2 genes, whose protein products interact and repress their own transcription. The stability of the period, PER, and cryptochrome, Cry, proteins are regulated by parallel E3 ubiquitin ligase pathways. Clock and BMAL1 also regulate the nuclear receptors Rev-ERB alpha and Rev-ERB beta, encoded by NR1D1 and NR1D2 respectively, which rhythmically repress the transcription of BMAL1 and NFIL3, which encodes nuclear factor interleukin-3 regulated, that is driven by the activators retinoic acid-related orphan receptor alpha, ROR alpha, and ROR beta, encoded by RORA and RORB respectively. NFIL3 in turn represses the PAR-BZIP factor DBP, D-box binding protein, to regulate a rhythm in the ROR nuclear receptors. These three interlocked transcriptional feedback loops represent the three major transcriptional regulators of the majority of cycling genes. Different combinations of these factors generate different phases of transcriptional rhythms, as exemplified by the graph top right, showing the RNA profiles of DBP, PER2, CRY1, and BMAL1 in the mouse liver. Additional rhythmic output genes, so-called clock-controlled genes or CCGs, are transcriptionally regulated by the three loops acting on E-boxes, RevDR2, and ROR binding elements, ROREs, and D-boxes in the regulatory regions of target genes. AMPK, 5' AMP activated protein kinase, CK1, casein kinase 1, CRE, CAMP response element, FBX, F-box protein, SCF, SKP1 choline F-box protein, SRE, serum response element, UB, ubiquitin. Box 2. Human genetics of clock genes. Validation of the core circadian transcriptional mechanism elucidated in rodents has come from the Mendelian disorder familial advanced sleep phase disorder, FASPD, in which the timing of sleep-wake cycles in affected individuals are shifted several hours earlier than normal. Dominant mutations in the genes that encode period 2 PER2 and the serine threonine kinases casein kinase 1 delta CSNK1D have been shown to be linked to FASPD and to be causative in humans. In large-scale human genome-wide association studies, GWAS, for fasting glucose levels and increased risk of type 2 diabetes mellitus, common variants in the MTNR1B locus, which encodes melatonin receptor 1B, and CRY2, and in CRY2, which encodes cryptogram 2, have been documented. Melatonin signaling is a major output pathway of the suprachiasmatic nucleus, SCN, in mammals, and the levels of melatonin are circadian, with high levels during nighttime and low levels during daytime, reflecting a physiological signal of darkness. The MTNR1B mutations have been proposed to act in pancreatic islets to impair insulin secretion, but this hypothesis remains a subject of debate. Recently, large-scale GWAS for morningness and sleep timing preference chronotype have identified several genes known to be associated with circadian rhythms in animals. These genes include RGS16, a regulator of G protein signaling that is enriched in the SCN and has a long period phenotype with a loss of function mutation. The core clock gene PER2, VIP, which encodes vasoactive intestinal polypeptide, a neuropeptide in the SCN involved in oscillator coupling, HCRTR2, the receptor for hypocretin and orexin that is linked to narcolepsy in humans and dogs, and FBXL3, an F-box protein that is the recognition domain for an E3 ubiquitin ligase complex that degrades the Cry proteins. GWAS using sleep duration as a phenotype have identified a few loci, PAX8, VRK2, and UFL1, but their relation to sleep mechanisms remains obscure. Overall, it is reassuring that numerous GWAS hits are associated with genes with known circadian and sleep functions, thus fortifying the notion that circadian transcriptional mechanisms are likely to apply to human biology. Figure 3. The circadian system of the mouse liver. A. University of California, Santa Cruz, UCSC, Genome Browser View of Chromatin Immunoprecipitation, followed by sequencing, ChIP-seq, profiles of circadian transcription factors at the PER1 gene at 6 circadian times of the day, 0, 4, 8, 12, 16, and 20 hours. The colors of the wiggle plots of ChIP-seq occupancy indicate the following transcriptional regulators: BMAL1 blue, clock green, PER1 orange, PER2 gold, Cry1 red, and Cry2 pink. KO indicates a ChIP-seq sample from a knockout mouse for each transcriptional regulator. B. Heat map views of genome-wide DNA binding for BMAL1, clock, PER1, PER2, Cry1, and Cry2 measured over 500 base pairs fragments encompassing the binding sites. Each peak in the genome is represented as a horizontal line ordered vertically by signal strength. 6 time points are shown beginning at CT0 and ending at CT20 from left to right. Knockout K mouse control is shown on the far right of each panel. The number of peaks in the genome is indicated at the bottom of each panel. The blue-red gradient indicates the coverage for all binding sites in the genome. The number of binding sites, N, in the genome are indicated at the bottom of each heat map. C. Chomsky diagram showing the six-way overlap of BMAL1, clock per 1, per 2, cry1, and cry2 peaks in the genome. The red circle in the middle represents the overlap of all six factors. Lighter shades of red, orange, and yellow represent fewer overlaps of subset. The boundaries for each protein are color-coded. BMAL1 blue, clock green, per 1 orange, per 2 brown, cry1 red, cry2 pink. And the areas of each domain are proportional to the number of binding sites. Figure 4. Whole transcriptome RNA sequencing analysis of circadian gene expression in the mouse liver. A. Heat map view of intron left and exon right RNA cycling genes. Each gene is represented as a horizontal line, ordered vertically by phase in hours indicated at the top. 12 time points are shown beginning at the circadian time 0, CT0, and ending at CT44 from left to right. N indicates the number of cycling genes in each category. B. Venn diagram of intron and exon cycling genes showing only 22% overlap. C. The phase distribution of cycling genes. The phase of each transcript rhythm is represented in a dot plot, top, and histogram plot, bottom. D. Heat map view of cycling RNA polymerase II 8WG16 occupancy across the genome of the liver with a peak occurring at night time. Part A and Part B. Okay. Figure 5. Circadian chromatin states in the mouse liver. A. University of California Santa Cruz Genome Browser view of histone methylation and acetylation at the per 1 gene at 6 circadian times of the day, 0, 4, 8, 12, 16, and 20 hours. The colors of the wiggle plots of chromatin immunoprecipitation followed by sequencing ChIP-seq signal indicate the following. GMAL1, BMAL1 occupancy, dark blue, monomethylation of lysine 4 at histone H3, H3K4ME1, red, H3K4ME3, pink, H3K9AC light blue, H3K27AC orange, H3K36ME3 light green, and H3K79ME2 dark green. B. Binding profiles of RNA polymerase II, 8WG16, and H3K4ME3 at the transcription start site TSS plus minus 3 kilobases in 12,680 expressed genes, first column, 8,945 unexpected genes, second column, 1,371 intron cycling genes, third column, and 5,839 non-cycling genes, last column. Figure 6. Circadian transcriptional landscape in the mouse liver. Histograms showing the phase distributions of each factor as a function of time of day. AC, acetylation. CBP, CREB binding protein. CRY, cryptochrome. ME, methylation. NPAS2, paralog of clock. Per, period. RNAP2, RNA polymerase II. SIR5P, phosphorylation on SIR5, reproduced with permission from reference 71 AAAS. Figure 7. Circadian transcriptional regulation at enhancer and promoter sites during the clock BMAL1 activation phase during the daytime. Clock and BMAL1 act as pioneer transcription factors at enhancer sites. Circadian recruitment and initiation, as seen by RNA polymerase II SIR5 marks of RNA polymerase II occurs at both distal enhancers and proximal promoter sites. However, the circadian regulation of posing factors, negative elongation factor, NELF, and DRB sensitivity inducing factor, DSIF, and pause release factors, positive transcription elongation factor B, PTEFB, remain to be determined. CBP, CREB binding protein, CRY1, cryptochrome 1, eRNA, enhancer RNA, GTF, general transcription factor, TF, transcription factor, TSS, transcription start site. Box 3. Four-dimensional regulation of the genome. It is well established that chromatin interactions occur in 3 dimensions. However, temporal changes in 3D genomic interactions, or in both space and time, the 4th dimension, are an emerging area, the 4D nucleome. Circadian cycles in chromosome organization have been observed using circular chromosome conformation capture 4C methods at the DBP locus, a well-known clock BMAL1 target gene. Although chromosome interactions surrounding the DBP locus were not reported to change during the circadian cycle, the frequency of long-range interchromosomal interactions did vary. Interchromosomal interactions were highest at the peak of DBP expression and were lowest at the opposite phase. When DBP expression was low, the DBP circadian interactome was dependent on BMAL1 expression because BMAL1 knockout cells had lower interchromosomal interactions, similar to that seen at the trough of DBP rhythms. 4C analysis of a BMAL1 bound enhancer upstream of the circadian gene NR1D1, which encodes the nuclear receptor REV-ERV alpha, also found that interactions were largely stable during the circadian cycle. However, a global analysis of cohesin and CTCF revealed that cohesin CTCF co-binding sites insulated cycling genes with different phases of expression, and that cohesin non-CTCF sites were associated with high amplitude circadian cycling. In addition, genome-wide chromosome conformation capture, Hi-C, combined with RNA sequencing expression, was used in human fibroblasts to search for circadian interactions, and the Laplacian analysis framework was implemented to quantify dynamic structure-function relationships in the genome. Using multicolor 3D FISH, clock and PER2, which have rhythmic and antiphasic structure-function dynamics, was shown to also have a correlated rhythm in spatial distance between them. Finally, using 4C to probe the H19 imprinting control region in human embryonic stem cells, a large interchromosomal interactome was identified. The interactome was organized by poly-ADP ribose polymerase 1, PARP1, and CTCF, and interestingly, active loci enriched for circadian genes were found to be recruited to repressed lamina-associated domains in the nuclear periphery. In serum synchronized HCT116 cells, there was a circadian rhythm of recruitment of circadian loci to the nuclear envelope that was dependent on PARP1 and CTCF. This recruitment was associated with the acquisition of demethylation of lysine 9 at histone K3, H3, A3K9ME2, repressive marks, and attenuation of transcription. Thus, circadian regulation of transcription also involves the recruitment and repression of circadian loci to lamina-associated domains in the nuclear periphery.